atoms of iodide added to tyrosine molecule, then you have T3. So the active thyroid hormones are either triiodothyronine or tetraiodothyronine, which is the thyroxine. The structure of T3 and T4 differ only by a single atom of iodine. And although T3 is more active than T4, almost all hormonal output of the thyroid gland is T4. So you are only secreting very small amount of T3. You are mainly secreting the major hormone which is coming out from the thyroid gland is T4. This problem of secreting less active form is solved by target tissue, which can convert the T4 to T3 because they have enzyme which is called 5' deiodinase, which can convert. We will take one iodine out and will convert T4 to T3. There's a third compound which is called reverse T3, and this reverse T3 is an inactive hormone. Okay, so if you have RT3, this is not an active hormone. Clear? So you have T4 and T3. Major output is T4, but the target tissues can convert T4 into T3 by an enzyme which is called 5' deiodinase. There is another type of hormone which is called reverse T3, and this reverse T3 is an inactive compound. It is not an active thyroid hormone. Now, if you look at the thyroid follicle, you would see that there are follicular cells. These follicular cells, they are arranged in a circular form to form a follicle of about 200 to 300 micrometers in size. Inside the lumen of the follicle, there is a colloid material which is stored there. And from this colloid material, which is mainly thyroglobulin, it's a glycoprotein, thyroglobulin, we are going to synthesize the thyroid hormones, the T3 and T3. T3 and T4 are going to be synthesized from thyroglobulin, which is stored as a colloid within the cavity of the follicle. So there is a cavity of the follicle and there is a lining epithelium, single layer of epithelium which is lining the follicle. So each follicle is lined by single epithelium. There is a cavity which is filled with colloid, and this colloid has thyroglobulin, which is a glycoprotein. From this thyroglobulin, which we have inside the cavity, we are going to synthesize the hormones, the T3 and T4. 7% of the triiodothyronine, and we produce 93% of thyroxine. So as we said, major output of the thyroid gland is thyroxine or T4. 93% of it is thyroxine. Only 7% of it is T3. But tissues are able to convert T4 to T3. The function of T3 and T4 are equally qualitative but differ in their rapidity and intensity. It means T3 is about 4 times as potent as T4, but less present in the blood and lasts shorter than T4. So T3 is 4 times more stronger, but it quickly disappears from the blood. T4 is less potent, but it stays for longer period of time in the blood. It does not get degraded as quickly. Okay. Now we are going to look at the steps of synthesis of the thyroid hormone, and this is very, very crucial. You need to understand how thyroid hormones are synthesized. This is something which you cannot forget out of this lecture. So you need to know each step of synthesis of the thyroid hormone. So first thing is that we have thyroglobulin, which is a glycoprotein synthesized by the follicular cells and secreted into the lumen of the follicular or the thyroid follicle. So when you are synthesizing glycoprotein, what you need? rough endoplasmic reticulum, which is synthesizing the protein. Then it goes to the Golgi apparatus where it gets modified into glycoprotein and packaged, and then it is secreted into the lumen of the follicle. So the first step is the formation of thyroglobulin, and thyroglobulin is rich in tyrosine. What you need to understand that each thyroglobulin has 70, each molecule of thyroglobulin has 70 molecules of tyrosine. So it's very rich in tyrosine as amino acid. It is synthesized by the rough endoplasmic reticulum of the follicular cells, packaged and converted into glycoprotein, and secreted into the lumen of the follicle, the follicular lumen, by the follicular cells. The second thing which you need to do is that we need to add iodine to these thyroglobulin. So what we in the blood have, as we eat our food, we have iodide in our food. This iodide goes into the blood from the GI tract, it goes into the blood, and it needs to be uh, concentrated within the follicular cells. So the follicular cells have a transporter which is called the sodium iodide co-transporter, which brings in 2 sodium with 1 molecule of iodide into the follicular cell. So it concentrates the iodide which is present in the blood into the follicular cell. Now, once this iodide enters into the follicular cell, it moves towards the, the apical portion of the follicular cell, or the portion which is next to the follicular lumen. Here we have an enzyme which is called peroxidase. This peroxidase will convert the iodide into iodine. So peroxidase, the thyroid peroxidase enzyme, is going to convert the iodide into iodine. And this iodine is going to get incorporated with the thyroglobulin, the tyrosine molecules of the thyroglobulin. So peroxidase enzyme not only oxidizes the iodide to iodine, it also get it incorporated to the tyrosine molecules of the thyroglobulin. If one iodine molecule gets attached to one tyrosine molecule, we call it monoiodotyrosine. If 2 molecules of iodine are going to attach to the tyrosine molecule, we call it diiodotyrosine. So peroxidase also catalyze the organification of iodine to tyrosine molecules in the thyroglobulin. So organification step is basically addition of iodine to tyrosine molecules on the thyroglobulin. So first, peroxidase is going to oxidize iodide to iodine. The second thing which it is going to do, it is going to add these iodine to the tyrosine molecules in the thyroglobulin, which is the glycoprotein present in the lumen of the follicular, in the lumen of the follicle. So this step is called organification. That means you are adding iodine to the tyrosine molecule, giving rise to monoiodotyrosine and diiodotyrosine. So you have now this MIT and DIT, which is monoiodotyrosine and diiodotyrosine. Now the third function of the peroxidase enzyme is called the coupling reaction. What is the coupling reaction? You would add one monoiodotyrosine to couple with another diiodotyrosine. So if one monoiodotyrosine couples with the diiodotyrosine, it will form triiodothyronine, which is T3. And if a diiodotyrosine is going to couple with diiodotyrosine, it will form tetraiodothyronine, which is T4. So peroxidase also is going to catalyze this reaction where you are going to form T4, T3, monoiodotyrosine and diiodotyrosine. But they are still attached to the molecule of the thyroglobulin. You have formed the T3 and T4, but they are still attached with the thyroglobulin molecule. So next what happens is that when you have the TSH attaching to the basal membrane of the follicular cells, there would be a pinocytosis of the thyroglobulin inside the cell. So they would take this thyroglobulin molecule, which has T4 and T3 attached with it inside the cell. And once it is inside the cell, the lysosomal enzymes will get attached with this thyroglobulin and will detach the T4, T3 from it. And this T4, T3 is going to go into the circulation. What is further left is the monoiodotyrosine and diiodotyrosine. So within the follicular cell, you have deiodinase enzyme, which will separate the tyrosine molecule and iodine. And this tyrosine will again gets incorporated to form the thyroglobulin, and iodide is going to be oxidized again and recycled into the follicular cells. If you have a deficiency of this enzyme, which is the deiodinase, then you would have deficiency of the thyroid hormones, because you cannot recycle the iodide and you cannot recycle the tyrosine molecule. So these are the steps of synthesis of the thyroid hormone. But we are going to go step by step and we are going to discuss these steps in detail. I've just given you an overview of how the thyroid hormones are synthesized. So again, these steps are listed here. You can see 1 to 8 steps are listed. Synthesis of the thyroglobulin, which happens in the rough endoplasmic reticulum and Golgi apparatus. Then you have the sodium iodide co-transport at the basal membrane, and this can be inhibited by perchlorate and thiocyanate. They can inhibit the co-transporter, so they would stop the iodide entry into the follicular cells. Then you have oxidation of iodide to iodine. This happens at the apical part of the follicular cells by an enzyme which is called the thyroid peroxidase. So this is the enzyme which does it. And you have this drug which we use to treat hyperthyroidism, and this is called the propyl thiouracil. So PTU means propyl thiouracil. This is a main drug which we use to treat hyperthyroidism. So it is going to block this enzyme, which is the thyroid peroxidase. So it will stop the oxidation of iodide to iodine. Then you have organification. That means the iodine need to be added to the tyrosine molecules, giving rise to monoiodotyrosine and diiodotyrosine. Again, this is happening at the apical membrane. The enzyme involved is thyroid peroxidase. Then you have a coupling reaction where monoiodotyrosine joins either diiodotyrosine, or a diiodotyrosine joins another diiodotyrosine, giving rise to T3 and T4. Again, this is happening at the apical membrane. Enzyme involved is thyroid peroxidase. Then once the TSH binds to the thyroid follicular cells, it would stimulate endocytosis of the thyroglobulin, which again happens at the apical membrane. Then once the T3, T4 are inside the follicular cell, it would be joined by the lysosomes to hydrolyze these enzymes from the thyroglobulin. And the enzymes involved are proteases. And then you have deiodination of the monoiodotyrosine, which has been engulfed, or diiodotyrosine, which has been engulfed in the follicular cell by deiodinase enzyme, and the iodine is recycled back into the follicular cells. So the first step, as we are not talking about, which is the synthesis of thyroglobulin. The second step is iodide trapping. That means you need to take the iodide from the blood and then put it into the follicular cells. So concentration iodide in the cell is 30 times more than the blood. So you can concentrate the iodide in the follicular cell more than 30 times what is the concentration in the blood. At maximal activity of the thyroid gland, the intracellular iodide may be more than 250 times more than the blood concentration of the iodide. And the TSH is important in iodide trapping. The pendrin, which is the chloride iodide counter transporter, is present here. You see at the apical border, while thyroglobulin can contain up to 70 tyrosine amino acids to which iodide binds and is secreted into the follicle. So we said thyroglobulin has number of tyrosines and to which iodine can bind. We need about 1 milligram per week of iodide is needed and for absorption from the intestine, and 1/5 of the circulating iodide is absorbed by the thyroid gland and the rest is removed by the kidney. So we need 1 milligram per week of the iodide. That's why you need your salt to be iodinated so that you can get at least 1 milligram per week. Otherwise, you may become hypothyroid because you don't have enough iodine in your food. So you need 1 milligram per week of iodide in your food. 1/5 of it is going to be taken up by the thyroid gland. The rest of it, 4/5, will be excreted by the kidney. One iodide is co-transported by 2 sodium. So you have the co-transporter here. One iodide is going in with 2 sodium molecules. So this will increase the sodium concentration in the cell also. So you need to remove the sodium. So you also have a sodium potassium ATPase, which is going to remove the sodium from the cell and will bring the potassium intracellularly. So you would have 1 molecule of iodide going in with 2 molecules of sodium. This is the main transporter at the basal part of the follicular cells. But to compensate this so that most of the intracellular sodium levels do not rise, you also have a sodium potassium ATPase pump, which is going to excrete the sodium out, bring the potassium in, so that there is hemostasis between the sodium and potassium intracellularly in follicular cells. No, you don't say this. You say that once 1 iodide enters, it will bring 2 sodium in. And to remove the extra sodium from the intracellular compartment, there is a sodium potassium ATPase. Yeah. So you don't want intracellular sodium levels to go up. Because the major ion intracellularly is potassium. So you need to take that extra sodium out, and that happens by there is an ATPase pump, sodium potassium ATPase pump, which takes out the sodium. Okay, the next step which happens is at the apical part of the follicle cells is oxidation. So we have the thyroid peroxidase, which oxidizes the nascent iodide to iodine. And then also thyroid peroxidase does organification, which means it adds iodine to tyrosine molecule, giving rise to monoiodotyrosine, thyrronine, and diiodothyronine. And these are then coupled as the coupling reaction is going to happen, giving rise to the T3 and T4. So then what happens is that the T3 and T4 are formed, but they are attached to the thyroglobulin and they are inside the follicular lumen. So when the TSH is going to come and get attached, it is going to result in penocytosis of the thyroglobulin inside the cell, and then you would have the protease enzymes in the lysosomes to lyse the thyroglobulin, releasing T3 and T4. Both of them would be released here, and they would go out into the blood circulation through the basement membrane. 3/4 of the iodinated tyrosine, monoiodotyrosine and diiodotyrosine, is not released from the thyroglobulin, and it is recycled uh, released in the cytosol of the thyroid cells by an enzyme which is called deiodinase. So the MIT and DIT are acted upon by the enzyme which is called the deiodinase. It will remove the iodine from monoiodotyrosine and diiodotyrosine, and you have the tyrosine also recirculated and iodide also recirculated. If you have deficiency of this enzyme, then you can lead to thyroid hormone deficiency. Again, this is the similar, much more comprehensive diagram. You see the first step is the formation of the thyroglobulin. Second step is the co-transport of the iodide. Then you have the oxidation. Then you have the organification reaction. Then you have the coupling reaction. Then endocytosis. Then the proteases are going to release the T3 and T4 from thyroglobulin, and these T3 and T4 will go into the circulation. Most of it would be T4, less amount of it would be T3. But in the target cells, we have 5' deiodinase, which can convert T4 to T3, which is the active compound. Similarly, what is happening in the follicular cells, that the thyroglobulin attached with the MIT and DIT, they are acted upon by deiodinase, which will release the iodine and also release the thyroglobulin, which can be recirculated. Now, the important thing in this picture is that the drugs which you can see here, one is the, you can see the propylthiouracil and methimazole. These are the main drugs which are used in hyperthyroidism, and you can see they can inhibit the thyroid peroxidase. So these drugs which are used in treating hyperthyroidism are going to inhibit the thyroid peroxidase, and that's why they can inhibit oxidation, they can inhibit organification, and they can inhibit what? Coupling reaction. So these three reactions can be inhibited by propylthiouracil and methimazole. The other thing is that propylthiouracil can also inhibit this enzyme, which is the 5' deiodinase. So it will also stop the conversion of T4 to T3 in the target cells. That's why it is a very effective drug which can be used for treating hyperthyroidism. If you have excessive amount of the thyroid hormone, this can be, you know, it can be stopped by the by this drug, which is the propylthiouracil. Okay, let's move forward. Okay, so now we have understood. Anybody has any questions regarding the synthesis? I think it's we have gone through it several times, so it should be very clear how the thyroid hormones are synthesized. But now these thyroid hormones, once they are released in the blood, they are not free. They are bound to certain proteins. They are transported after binding to certain proteins, and these proteins are called thyroxine binding globulin, or some amount of it also binds to albumin and then transported to the target organ. There's a very small amount of free thyroxine or very small amount of free T3. The rest of it is going to be transported by binding to the thyroxine binding globulin. Now this thyroxine binding, once T3 or T4 is bound to the thyroxine binding globulin, it is inactive. The only part of the thyroid hormone which is active are those which are free T3 or free T4. So free T3 and free T4 are active. The bound, to the globulin are inactive forms of the hormone, and they are slowly released from the globulin to act on the target tissues. Do you understand this? Now, this thyroid binding globulin is synthesized by the liver. Okay, so the liver is the major organ which is basically synthesizing this thyroid binding globulin. So if there is a problem with the liver, like liver failure, or you have a problem in the kidney like nephrotic syndrome where you are excreting a lot of protein out of the kidney, you would have lower amounts of thyroid binding globulin. So if you have lower amount of the thyroid binding globulin, your free T3 and T4 would increase. And if there is high levels of T3 and T4, they will have a negative feedback on TSH. So they would stop the synthesis of the thyroid hormones. So when you have hepatic failure or you have nephrotic syndrome, you would have lower amounts of thyroid binding globulin and you would have high levels of free T3, T4, which will ultimately affect the TSH and bring down the synthesis of the thyroid hormones. On the other hand, there are conditions where you have high levels of thyroid binding globulin, for example, in pregnancy, or you have the use of OCP or oral contraceptives where the estrogen is there, and estrogen is going to increase the thyroid binding globulin. So if you have high levels of thyroid binding globulin, the free T3 T4 would be less. So this would stimulate the TSH and the synthesis of the thyroid hormones will increase. So the total amount of T3, T4 in pregnancy and in use of estrogen will result in increased T3 and T4, but the person would still be euthyroid because the amount of the free T3, T4, which is the active form, is the same. But because you have high levels of thyroid binding globulin, you would have high levels of T3, total T3, T4, because it would result in lower free T3, T4, resulting in release of TSH and increase in the synthesis of the thyroid hormone. So the total amount of free and bound T3, T4 would be high, but the person would be euthyroid. That means he would not be hyperthyroid or hypothyroid, but he would be euthyroid. But if you count the total amount of T3 and T4, it would be higher. Do you understand this concept? These are some things which, USMLE step one people really like to ask. They want to know what happens in pregnancy to the thyroid binding globulin, what happens to the total levels of the thyroid hormones, what happens to the thyroid hormones in the hepatic failure, those type of questions. So you need to know about them. Okay. Okay. Now we are coming towards the regulation. The major regulation of the thyroid hormone synthesis is under the anterior pituitary. Anterior pituitary is the one which is going to release a hormone which is called the thyroid stimulating hormone. It is a glycoprotein of 28 kilodalton. It increases the thyroid hormone secretion, and it actually increases all these steps of thyroid hormone secretion. For example, it increases the proteolysis of stored thyroglobulin. It increases the iodide pump activity. It increases the iodination of tyrosine. It increases the thyroid gland size and the secretory activity of the thyroid gland. And it increases the number of thyroid cells with the change in shape from cuboidal to columnar. So TSH, if the TSH is high, it is going to not only increase the synthesis of the thyroid, all the steps of synthesis of the thyroid hormone, but it also is going to physically affect the thyroid gland. That means the size of the thyroid gland is going to increase and the number of there would be hyperplasia or the increase in the number of the follicular cells. So sometimes hyperthyroidism or hypothyroidism, whenever you have high amount of TSH, it would result in large thyroid gland. So whenever you have higher amount of TSH, not the thyroid hormone, we are not talking about hypothyroidism or hyperthyroidism. Whenever you have high levels of TSH in the blood, it will result in larger thyroid gland, which is called a goiter. So when you see a goiter, you always think about TSH is high. It could be TSH is usually high in hypothyroidism, but sometimes in hyperthyroidism also there's a substance like similar to the TSH, which is called the thyroid immunoglobulin, which has stimulating immunoglobulin, TSI, which has a similar structure to the TSH. So like in Graves' disease, which is hyperthyroidism, but you have large thyroid gland because it works like TSH. So TSH, whenever you have high levels of TSH, you would have larger thyroid gland. But TSH also increases the all the steps, activity of all the steps of synthesis of thyroid hormones, which we have talked about. Iodine, trapping iodine, organification, oxidation, coupling reaction, hydrolysis of the thyroid hormone. All the steps of the synthesis of the thyroid hormone are increased by the TSH, which is basically produced by thyrotropes from the anterior pituitary gland. Yeah, yeah. Before this one. So we said that once the thyroid hormones are released, the thyroid hormones are mostly bound to a globulin, which is called thyroxine binding globulin, which is the TBG. Some amount of the thyroxine hormones are also bound to albumin, but mostly they are transported bound to the thyroglobulin. The free T3 and T4 are in very small amount, but they are the ones which are the active hormones. When T3 and T4 are bound to the TBG, they are inactive. So slowly they are released from the TBG when they reach their target organ, and the free T3, T4 are the active hormones. Now, in conditions, TBG, as I said, is produced or synthesized by the liver. So in conditions where the liver is damaged, TBG is produced in lesser amount, or in a condition like nephrotic syndrome where proteins are lost through the urine, you are losing a lot of these globulins. So you have lower amount of TBG. When you have lower amount of the thyroid binding globulin, you would have higher levels of T3, T4, which is going to affect the pituitary gland to decrease the TSH. And as the TSH is going to go down, the synthesis of the thyroid hormones is also going to go down. On the other hand, conditions like pregnancy or in conditions where you have high levels of estrogen, for example, you take oral contraceptives which have a lot of estrogen, this will increase the amount of thyroglobulin, thyroid binding globulin in the blood. And as there would be larger amount of the thyroid binding globulin there, the levels of free T3, T4 would be lower. And as a result, it would stimulate the pituitary gland to release TSH, and you would have higher synthesis of thyroid hormones. So the total amount, the free and bound, T3, T4 in pregnancy would be higher because you are stimulating the thyroid gland to, to produce more hormone. So although the person is euthyroid, that means the uh, the the effect of these hormones is normal because the amount of free T3, T4 is not higher, you would have larger amount of total T3, T4 bound and unbound because it has stimulated the thyroid gland to produce more thyroxine. Okay, is it clear? This slide we have already talked about that TSH is the major hormone which is responsible for uh, increasing the synthesis of the thyroid hormones, but it also affects the size and of the thyroid gland and number of the follicular cells in the thyroid gland. So hyperplasia and hypertrophy of the gland is because of TSH. So when you have high levels of TSH, you have larger gland, which is goiter, and you also increase the synthesis of thyroid hormones. Now, if we look at this diagram, diagram is actually showing you the full regulation. There is a hormone which is released from the hypothalamus, which is called the thyroid releasing hormone. And this thyroid releasing hormone acts on the anterior pituitary, where you have thyrotropes. And these thyrotropes release TSH, which stimulate the thyroid follicular cells to synthesize and secrete T3 and T4, which affects the target cells. Now, this T3, T4 can have a negative effect directly on the anterior pituitary, where they would downregulate the receptors of the TRH. So the receptors of TRH are downregulated in anterior pituitary. So what would happen? Even if you have the TRH, the anterior pituitary are not going to secrete the TSH. So there is a negative feedback of free T3, T4 to affect the anterior pituitary, where they are going to downregulate the receptors for the TRH. T3, T4 can also directly inhibit hypothalamus to secrete the TRH. So there are two negative feedbacks. One to the anterior pituitary, where they are down-regulating the receptors for TRH. The other negative feedback of T3, T4 is to directly inhibit the hypothalamus to secrete the TRH. So when you have high levels of T3, T4, it is going to affect the TSH, it is going to affect the anterior pituitary, and it is going to affect the hypothalamus to decrease the secretion of TRH, and to decrease the, at the anterior pituitary, to decrease the receptors of TRH, resulting in that anterior pituitary is going to decrease the amount of TSH which is being produced from the anterior pituitary. The other hormone which can actually inhibit the anterior pituitary to secrete TSH is somatostatin. So somatostatin can also directly inhibit the anterior pituitary to secrete TSH. In this diagram, you are also seeing something which is called TSI. TSI is normally not produced by the body. It's the thyroid stimulating immunoglobulin. It is only produced in an autoimmune disease which is called Graves' disease. And this TSI has a structure which is similar to TSH. So it binds to the receptor of TSH. So it has the same action which the TSH has. So what does TSH do? Increase the size of the gland, increase the number of the cells, follicular cells, increase the synthesis of the hormones. So TSI is the autoantibody which is produced in a disease which is called autoimmune disease, which is called Graves' disease, and it stimulates the thyroid follicular cells like TSH and increase the production of the thyroid hormones. Okay, so in this case, when you would have large number of T3, T4, activated by TSI, it would, although inhibit the anterior pituitary to release the TSH, but you still have this antibody which is continuously activating the thyroid hormone. The negative feedback is working. You have lower level of TSH, but you have high levels of these antibodies which are continuously produced by the body in this disease, which results in hyperthyroidism. Do you understand this concept? Okay, so this is what is basically shown here. The hypothalamus releases TRH in the median eminence. This TRH is brought to by the microcirculation to bind to the receptors on the anterior pituitary, the thyrotrope cells. Activation of the THR receptor increases the phospholipase C and the second messenger system, which includes calcium and DAG, resulting in the secretion of TSH. Now, once the TSH is secreted from the anterior pituitary, it binds to the TSH receptor on the basal lamina of the thyroid cells, activating cyclic AMP and activating the protein kinase A and phosphorylation of the several protein. This results in thyroid hormone secretion and production. So if you look at the TRH, it is working through the phospholipase C and calcium second messenger system, while you are seeing the TSH when it binds to the its receptor, it is working through the cyclic AMP and protein kinase A as the second messenger, resulting in the synthesis of the thyroid hormones. Now, as we said, that now we are moving towards, we have talked about the regulation, we are moving towards the functions of the thyroid hormone. What does thyroid hormone do in the body? That's also very important. And then finally, we are going to move towards the clinical aspects. You can take a break of 5 minutes before we start the actions, and then we are going to move towards the clinical aspects. Any questions? I can answer the part which we have done.
You were mentioning TSH. As idols of HS, we're going to have a large thyroid gland. That will cause hypothyroidism. What were you saying?
No, we are not. We high levels of TSH is going to cause large thyroid gland.
Okay.
But it doesn't, it usually is associated with hypothyroidism, not hyperthyroidism. Hyperthyroidism can cause large gland, but because of a thyroid stimulating immunoglobulin, which has a similar structure to TSH.
Okay.
Okay, but usually it's the hypothyroidism where you would have a large gland because you have high levels of TSH. But in Graves' disease, which is hyperthyroidism, you still have a large gland. So people say, okay, hyperthyroidism should have lower level of TSH, but why they have a large gland? It's because they secrete thyroid stimulating immunoglobulin, which has a similar structure to TSH.
I get it. Thank
Doctor, I still don't get why when we have increased TBG,
Mhm.
we
TBG.
yeah TBG,
Mhm.
we will have a decrease in the T3 and T4 free form. Then we will have an increase in the TSH, right?
So when you have high level of TBG,
Yes.
like in pregnancy
Yeah.
and estrogen, when you have excessive
Yeah.
amount of estrogen, the amount of free
Yeah. The
T3
free form.
free form would be decreased. When you would have large amount of TBG produced, it will bind to all the thyroid hormones which are being
So we
produced
will have a decrease in the
in
free form.
free form. So when you have a decrease in free form, it is the free form which is
Going to do the feedback, the negative feedback.
Yeah. So when the pituitary gland would detect there is low level of free T3, T4, it would start to secrete TSH.
But why he's why he's euthyroid?
I will tell you why. So it would start to produce, T3, T4.
Yes.
Okay. And because you have large amount of, TBG being produced,
Yes.
it would still bound to the most of the thyroid hormones which are produced by the thyroid gland. And the amount of the free T3, T4 would be normal.
I still don't.
You take
Like the extra amount of T3, T4 is
already bound to
bounded.
extra
I
TBG.
mean, the high level of T3 and T4 is in the bounded globulin.
It is
So
in,
it's fine.
the
As
person
long-
would be euthyroid, so they would not show us
Like
symptoms
yeah.
of
So you
hyperthyroid.
mean as long as the T3 and T4 is in the bounded form?
Doesn't
It
matter.
will not. It doesn't matter
The
because it
only
will
one
not give
matters
you
is the
effects.
free ones.
It's the free T3,
Okay,
T4
okay,
which is
okay,
going to give
okay.
you the effect.
Now I get it. Thank you.
Okay, doctor, I have a small question. So now for the TSH, if it's very high, it doesn't have like a negative direct feedback. I mean, when it's so high, it does not like inhibits the TR the
TRH?
Yeah.
TSH
So that is-
does not
yeah,
do anything.
That is why that the patient have like a large thyroid gland because like it does not go down in any case.
Yeah. So
only with
the TRH anterior pituitary as well as hypothalamus are only associated with T3, T4 levels.
Yeah.
So if your T3, T4 levels are low, the TRH and TSH will continuously be produced.
Yeah.
If they become high, they would stop.
Yes. Okay. Thank you.
Again, there's a question about TBG.
Mhm.
In liver failure and similar conditions where less TBG is produced, does that also appear euthyroid?
No.
What are the effects when there is low TSH?
Mhm.
Are they the opposite of high TSH?
Low TSH.
Mhm.
So when you have low TSH, you are actually hyperthyroid because you have high levels of T3 and T4. That would cause low level of TSH. Yeah.
Is there a treatment for the cases associated with thyroxine binding globulin, or are they treated by PTU levothyroxine?
Uh, the thyro cases associated with
The
TSI,
TBG.
TBG, we don't need to treat. We need to treat the condition which results in lower level of TBG like liver failure or renal disease. We don't need to treat, the thyroid hormones. Yeah.